slide no. So continuing the classification of bacteria. So spherical shaped organisms are called cocci. Singular is coccus and plural is cocci. Singular is coccus and plural is cocci. So there are 5, hold like this, 5 cocci which cause infection in man. Staphylococcus, gram-positive cocci arranged in clusters. Cocci arranged in clusters. It looks like bunch of grapes. Staphylo means grapes. Okay. Streptococci, gram-positive cocci arranged in chain. Gram-positive cocci arranged in chain. They look like serpent. Streptococcus means serpent. Streptococcus means serpent. The third one is pneumococci. Hold like this. They are lancet shaped, broader and facing each other. 2 cocci enclosed in a single capsule. 2 cocci enclosed in a single capsule. Then all next 3, first 2 are cocci, which are one is in clusters, staphylococci, one is chain, that is streptococci. Pneumococci, gonococci and meningococci are diplococci. They are in pairs. Streptococci is pneumococci is lancet shaped or flame shaped. Hold everybody like this, broader and facing each other. And this is meningococci, half moon, plain surface facing each other. Gonococci, kidney shaped, and the concave end facing each other. Staphylo, strepto, pneumo, gono, meningo. Staphylo, strepto, gono, meningo, pneumo. Staphylo, strepto, gono, meningo, pneumo. So now moving on to the bacilli. Bacilli are rod-shaped organisms which which are 3 micron in length and 0.3 micron in breadth. 3 micron in length, 0.3 micron in breadth. They are classified depending on 3 criteria. One is gram staining. The second one is they are classified on 3 criteria. One is gram staining, second one is spore forming capacity, the third one is acid fastness. Now acid fast organism, they don't get decolorized with acid and alcohol. They don't get decolorized. These organisms do not get decolorized with acid and alcohol. The example of acid fast and alcohol fast organisms are Mycobacterium tuberculosis, and Mycobacterium leprae. You heard of tuberculosis? Anybody? No, sir. So usually the patient comes to hospital with loss of weight, evening rise of temperature. Sometimes there will be coughing of the blood. The sudden loss of weight, coughing of the blood, and evening rise of temperature. Leprosy, all the beggars, what you see outside the temple, Gurudwara and masjids are leprosy patient. This clawed face, no eyebrows, and saddle shaped nose. Usually they are blind, so they take up blood begging. So leprosy infects the nerves and the skin. It's called neurodermatitis. Now spores are highly resistant dormant stage of bacteria. Highly resistant dormant stage of bacteria. Spores are highly resistant dormant stage of bacteria. Highly resistant dormant stage of bacteria. They formed in unfavorable condition. They formed in unfavorable conditions. When the conditions are not favorable, they are formed. Example are Clostridium tetani, Clostridium botulinum, Clostridium welchi. These are also called anaerobic organisms because they grow in absence of oxygen. They grow in absence of oxygen. So spores are formed in absence of oxygen. Spores which are formed in presence of oxygen are anthrax bacilli. Anthrax bacilli. It's a zoonotic disease. The first zoonotic disease you should remember is rabies. Man get infected by the bite of the dog. It's a dead end disease. So don't neglect the dog bite. Immediately you should report to the hospital and take anti-rabies vaccine. Anti-rabies vaccine. It's very important because once you get infected with rabies, nothing can salvage you. Dead end disease. Now these are acid-fast organisms and these are spore-bearing organisms. Spores are highly resistant resting stage of bacteria. Spores are highly resistant resting stage of bacteria. So repeat with me, acid-fast organisms are, everybody, acid-fast organisms are Mycobacterium tuberculosis and Mycobacterium leprae. Not leprosy, leprae. It causes leprosy. Disease is called, infection is called leprosy. The organism is called leprae. Leprae. And spores are, everybody, spores are highly resistant resting stage of bacteria. And example of aerobic spore bearers, oxygen which is formed in presence of oxygen. In presence of oxygen. In presence of oxygen is anthrax. Anthrax bacilli. Man get infected by inhalation of the spores. Inhalation of the spores. It's a zoonotic disease. Diseases of sheep. So we are in close contact with the sheep. We use wool and we eat sheep meat, mutton, and the spores are present there and we get contact with the skin, it gives a malignant pustule. When you breathe, it's called as wool sorter's disease, pneumonia, and it can also give rise to hemorrhagic enteritis, infection of the intestine. Now moving on to the gram staining. Today we are doing gram staining in the practical because the PowerPoints are now not working. And what is gram staining? It was discovered by Hansen Christian Graham. So everybody repeat with me. It was discovered by Hansen Christian Graham. Differentiates between two group of organisms. Gram positive organisms and gram negative organisms. Gram positive organism look violet in color and gram negative organism look pink in color. Gram negative organism look pink in color. Everybody, gram positive organism look violet in color and gram negative organisms look pink in color. Okay. Now it is a 4-step procedure. How many steps? 4 step procedure. We write gram here. Smear. Okay. Cover the smear with gentian violet for 2 minutes. Cover the smear with gentian violet for 2 minutes. Wash with water. Then add second step. Second step, mordant. Mordant is iodine, grams iodine. Mordant is, it fixes dye to the cytoplasm. It fixes dye to the cytoplasm. The third step is wash with water. Decolorize with alcohol or acetone, drop by drop until faint color remains. Counter stain with concentrated carboxyl. Now, this is because of difference between the gram positive and gram negative cell wall. Gram positive look retain the primary stain violet, and gram negative organism give away the primary stain and take up the counter stain. Okay. The procedure you write down. The procedure of gram stain. Principle of gram staining. The staining is done to differentiate between gram positive and gram negative organism. Gram positive organism and gram negative organism. Procedure. Cover the smear with gentian violet for 2 minutes. Gentian violet, any violet you can use, methyl violet, crystal violet, but we usually use gentian violet. Cover the smear with gentian violet for 2 minutes. Wash with water. Add mordant, grams iodine, grams iodine, for 1 minute. Wash with water. Decolorize with alcohol, drop by drop until a faint color remains. Counter stain with counter stain with dilute carbalfuchsin. Wash, blot dry, see under oil immersion microscope. Wash, blot dry, see under oil immersion microscope. So gram positive organism look violet in color, gram negative organism look pink in color. So why some organism look violet in color and why some organism look pink in color? These are explained by Gram's postulate, mechanism of Gram reaction. Number one, right, mechanical theory or cell wall theory. Mechanical theory or cell wall theory. Gram positive cell wall is 40 times thicker than gram negative cell wall. How many times? 40 times. 40 times thicker than gram negative cell wall. How many times?
40.
How many times? This is 40 times thicker than gram negative cell wall. Okay. Now, but gram negative cell wall has got outer covering which is made up of lipoid. It's very complex. It has got lipoid layer outside the cell wall. When you add alcohol, decolorizing agent, it gives away the primary stain diiodine complex. Gives away the diiodine complex. Write down. Mechanical theory, 40 times thicker, and gram negative cell wall is made up of lot of lipids, while decolorizing with acetone or alcohol. Decolorizing with alcohol, the alcohol dissolves the lipid in the cell, gram negative cell wall. It gives away the diiodine complex. One is mechanical theory, or number two is electrical theory, mechanical theory. Is it clear?
Yes, sir.
Second is electrical theory. Second one is gram positive cytoplasm. Gram positive cytoplasm is made up of acidic pH and more affinity or more binding towards basic dye. What we use is all basic dyes. They have more attraction, more affinity towards basic dye. Compared to gram negative, gram positive are more acidic in nature. Written down. Compared to gram negative, gram positive cytoplasm is acidic in nature, and they have more affinity towards basic dye. Gram positive cytoplasm. Written down. Gram positive cytoplasm. Written down. Are acidic in nature, more acidic than gram negative. More acidic than gram negative. They have more affinity, love, attraction towards basic dye. Shall I repeat once again? First is mechanical theory. First gram positive cell wall is 40 times thicker, but gram negative is made up of lot of lipids. Alcohol dissolves the lipids, the iodine complex comes out. Gram positive cytoplasm is acidic, and more attraction towards basic diet. More attraction. The third theory is chemical theory. There are two chemical theory: magnesium ribonuclease and teichoic acid. Magnesium ribonuclease and teichoic acid present in the magnesium ribonuclease, teichoic acid, magnesium ribonuclease and teichoic acid present in the gram positive cell wall are absent in gram negative. They are also responsible for retaining the primary stain. Once again I'll repeat. One is mechanical theory, that is gram positive cell wall is thicker, gram negative thin wall is thinner, but gram negative cell wall is more complex, it contains lipid. Alcohol is dissolvent, it gives away the iodine complex. Number 2, gram positive cytoplasm acidic, more attraction towards basic diet. The third one is magnesium ribonuclease and teichoic acid, which makes to retain the primary stain. Okay. Now moving on to the example of gram positive and gram negative organism. Now first 3 cocci are gram positive. First 3 cocci are gram positive. Staph, strepto and pneumo are gram positive. Staphylococci, streptococci, pneumococci are gram positive. Staphylococci, streptococci, pneumococci are gram negative. And gonococci and meningococci are gram negative. Staphylococci, streptococci, pneumococci are gram positive. Staphylococci, streptococci, pneumococci, SSP are gram positive. They look violet in color. Gram negative are gonococci and meningococci. They look pink in color. Now moving on to the bacilli. Bacilli are rod-shaped organisms. Okay. Example of Corynebacterium. Corynea means it looks like a thorn. It gives rise to diphtheria. Then lactobacilli present in the milk. Then listeria. Listeria monocytogen gives rise to meningitis. Then anthrax and Clostridium. These are all gram positive. Once again, I'll repeat in nutshell total procedure. Keep the smear facing upwards. Cover the smear with gentian violet. Wash with water. Add mordant which fixes dye to the cytoplasm for 1 minute. Wash with water. Decolorize with acetone or alcohol. Counter stain with dilute carboxylic. Wash, blot, dry and see under oil immersion microscope. Gram positive organism look violet in color and gram negative organism look pink in color. So why some organism look violet in color? Why some organism look pink in color? Because of the mechanism, postulates of gram. One is mechanical. Cell wall is thicker. Gram negative has got more lipids. Alcohol dissolves the lipid in the cell wall. Number 2, acidic nature of the gram positive cell and it attracts the basic dye. Number 3, magnesium ribonuclease and teichoic acid. Example of gram positive cocci are staphylococcus, streptococci and pneumococci. Gram negative cocci are meningococci and meningococci. Gram positive bacilli are corynebacterium, lactobacilli, listeria, anthrax and clostridium. So rest are all gram negative. And one more you can add is actinomycetes. Actinomycetes are all gram negative. You maintain a separate notebook for microbiology. Okay. First question I will give you today. You write that and bring and show me. First question is write the diagram of microscope. Write the diagram of microscope and describe various parts of microscope. How do you classify? Second question. How do you classify bacteria depending on the shape? First question you want to repeat? Second. Classify bacteria. Classify bacteria depending on the shape, size and arrangement. Shape, size and arrangement. Classify bacteria depending on shape, size, arrangement. 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 Third question. Principle of gram staining, procedure of gram staining. Example. Principle of gram staining. Principle, not principal. Principle of gram staining. Principle of gram staining and mechanism of gram staining. And why some organisms are gram positive and why this is described as Gram's postulate. What are Gram's postulates? Gram's postulate, POSTULAT, postulates, Gram's postulates. Example of gram positive and gram negative organisms. So only 10 people can see, rest of them will share the video in your WhatsApp group. So we can't accommodate all. Now move to the practical hall.